Hello, good morning, good afternoon, good evening, or even good night, depending from where you are connecting today to this webinar. My name is Federico Martinón-Torres. I'm a pediatrician and clinical researcher, and I will be your chairman, your host, your moderator for the next hour, when we will have a unique opportunity to discuss about a very important issue, how it is difficult vaccine targets, including COVID-19, but with the unique opportunity to do that discussion with a person, with a professor that, I usually don't get nervous when I give a talk, or even less with a webinar where you don't have the face-to-face interaction, but today I'm thrilled. I have this emotion of having the opportunity to share the bill with someone like Professor Stanley Plotkin. Actually, if you don't know who is Professor Stanley Plotkin, this webinar is not for you. Probably this is not something that you should be viewing now. Good morning or good afternoon, Stan. How are you?
Muy bien, gracias.
Thank you very much, Stan, for accepting this challenge and for joining us today in this webinar. I am really honored, and I really thank you on behalf of all my colleagues in Hospital Clínico Universitario de Santiago and my research group. And on behalf of the audience, we have almost 1,000 people registered to today's webinar. We will know later how many of them actually have connected to the webinar, but I think that we will have an hour that will be very, very interesting and exciting, and we will have the chance to discuss with you and try to answer the audience regarding the situation we are now facing about the development of a vaccine against COVID-19, and having the opportunity to get your insight, being as you are one of the most important experts in the world of the vaccine. So again, thank you, Stan, and welcome to this webinar. I also want to thank, of course, all the people that has registered to the webinar. And also I'd like to thank the institutions endorsing today webinar, and of course also Pfizer for giving an unrestricted support to provide all the IT facilities to have this webinar we are having at this moment. I hope everything goes fine from a technical perspective. You know that you can access this webinar in English or in Spanish. You only have to click on the right language you want to listen to us. And also you can ask any question using the specific box for questions. And after my talk and Professor Plotkin talk, we will have some time to try to deal with all your questions. So said this, I think we are going to get started and go directly to the matter of this webinar. Well, when we discuss the possibility of doing this webinar, we thought that indeed, this is myself and this is my team. I'm a professor of pediatrics at the University of Santiago, and in the left corner you can see my research group, GenBip, and also on the right, the Department of Pediatrics, where I'm the head, and I have the honor of the challenge of being the head of the Department of Pediatrics of the Hospital Clínico Universitario de Santiago here in Spain. As said, when we planned this webinar, we were, as we all are, facing a difficult situation. We are all facing a situation where a vaccine is badly needed. And usually it's important to reflect what can we learn from what has been already done or what is currently being doing, to really know better how to deal with a new problem like COVID-19 is. For that reason, we thought that no one could do that better than Professor Stanley Plotkin, and knowing how, and despite I know how busy he is, his friendship and his eagerness to collaborate with anyone that requests his help has made this possible. Also, these are the institutions that have endorsed this webinar: my own institutions, my hospital, my department, and my research group, and as said, also the collaboration sheet from Pfizer to make the technical part of this webinar possible. The basis, the background for this was exactly to see where we are in the in the development of vaccines against COVID-19, but also to see what can we learn from past and current lessons in this career to achieve as soon as possible a safe and effective vaccine against COVID-19. I will take only a few minutes to reflect before giving the floor to Professor Plotkin on vaccination despite COVID-19. This is my potential conflict of interest. I won't be speaking about any specific brand or vaccine, but I work with most of the vaccine sponsors as head of a vaccine clinical trial unit. Well, we could think that we are in a good moment for vaccines in the sense that, well, everybody is willing to have a vaccine to solve this problem, the COVID-19 issue, and then we could think that everything is now solved in terms of vaccine hesitancy or issues related to vaccine perception. I like this sentence, which is not mine. I don't know exactly who noted it, but says, for those who wanted a world without vaccines, here is the world without one vaccine. And we could think that, well, it will take more or less time to achieve a vaccine against COVID-19, but probably the problem of vaccine hesitancy is solved. Well, at all. This scary paper recently published by Johnson at Nature just make us reflect how even in the current situation, the anti-vaccine lobby, despite the low number they are spreading their anti-vaccine messages, and their influence is quite striking. Here we can see in different colors the anti-vaccine lobby in red, and despite in numbers they are less than the pro-vaccination people, their interaction and the intricacy of their interactions is much more important in the population that is undecided. So we can't at all, even in the current situation, lower our guard regarding the anti-vaccine lobbies and the spread of their anti-vaccine messages. Not related to that, but more to the actual situation of COVID-19, we are facing and we are observing worldwide a decrease in the vaccine coverage with routine vaccines, not related to vaccine hesitancy probably, or even less to anti-vaccine groups, but more relating to the difficulty to access the vaccination system, or even the doubts or the fears to go to the vaccination point. This can become a true important issue. We can see here the concrete data released by CDC, but this is being seen in several other countries worldwide. So it's very important that we resume as soon as possible routine vaccination, and we make our most important effort in order. We have to go back to containing measures, lockdown situation, to ensure that the vaccination is maintained. Because if we stop vaccinating, the other disease different to COVID and even as dangerous, if not more than COVID, will come back. Also, there are other reasons to consider vaccination, because we know, although the information is limited, that COVID can come with other pathogens, that co-infection can exist. We still don't know much about the concrete proportion of other microorganisms that can, let's say, work together with COVID, and which is the influence in outcome, in severity, etc. But it's important to outline that some of them, as you can see in the figure, are well recognized and vaccine preventable pathogens. You can see in the first place how the Streptococcus pneumoniae, for which we have safe and effective vaccines, is present in many of the patients with COVID-19 described in this series. As said, we still don't know much about the concrete interaction and how important is the co-infection in the course of a COVID-19 disease, but I think that any vaccine preventable disease should be prevented, even if it is just to eliminate one other possibility of attending the medical system or of being exposed to COVID-19 itself. Furthermore, in this situation, and not having yet vaccines available against COVID-19, several groups are working trying to repurpose already existing vaccines to prevent or to partially prevent COVID-19 disease. Some of these examples have got great attention in the media, and we are talking about what many of you are familiar with, the heterologous effects of vaccines, or the effects that vaccines may have against other diseases different to those for which they have been conceived or they have been developed and tried. Actually, we are talking about BCG vaccine, we are talking about polio vaccine, we are talking about measles vaccine, live attenuated vaccines that have shown in other contexts through different mechanisms, prevention of immune amnesia in the case of measles, training of the innate immune system, like BCG, probably a pro-inflammatory response with the oral polio vaccine, whatever, but showing an extended benefit against other infectious diseases and even an impact on all-cause mortality. We can see here some of the drivers of these ideas. In the first picture above is Peter Abbey, who is one of the leads in the field of heterologous effects of vaccine. And in the picture below, you can see Carlos Martin, who is the inventor and the father of the new upcoming tuberculosis vaccine, the MTB vaccine, that is going to be also tried in the context of prevention of COVID-19. Well, but what actually matters us, what really concerns us, is to achieve a safe and effective vaccine against COVID-19. And I think the good news is that we have a unique and without precedent global commitment. All the stakeholders, the academia, the sponsors, the industry, all the possible agencies related to vaccination are working together in that same direction. And this is great news, and I think is one of the beauties or the good parts, if we can say so, of this SARS-CoV-2 pandemic. As a matter of fact, several candidates are now in the pipeline. We have almost more than 130 candidates, and some of them are already in the human phase. And as you know, there are different initiatives to accelerate the approval or the achievement of the final licensed product and to have vaccines available as soon as possible to actually deal and change the situation with regards to COVID-19. But these are the things that we will discuss with Professor Plotkin, and there are also other initiatives where, of course, again, Professor Plotkin is also very familiar, and he is part of the experts groups related to WHO and the R&D blueprint related to COVID vaccines. And also, as you are aware, there are initiatives like this Solidarity Vaccine Trial that will try to perform in a adaptive design, multi-country design, the test of the efficacy of the candidates that achieve the phase 3 stage in their development. Indeed, the challenge that the COVID-19 vaccines are facing are huge. We still don't know many things about the natural disease itself, and these are indeed hurdles and challenges for the vaccine development. Some of them, I'm sure, that Professor Plotkin will solve, or at least will give us hints to understand and to know where we are. We still don't know for sure if the immune response induced will be fully protective, I mean the immune response elicited by the vaccines, which will be the duration of the protection, the perfect antigenic target, which is the best correlate of protection. We don't know if these vaccines will provide or not mucosal protection. And very importantly, we need to ensure that any vaccine that is licensed against COVID-19 guarantees the safety of the vaccinee. And I think this very recent editorial from Nature Review, from Chandler, tell us some interesting thoughts about the importance also to coordinate all the efforts in the surveillance of the safety of the new and upcoming vaccines against COVID-19. We need to do an extra effort to do real-time global data exchange. We have to pull any report of IFIs. Probably we need to get ready for an ex-IFI, for an unforeseen potential adverse event following immunization with these new technology-based vaccines. Probably we have to consider syndromic surveillance, and also, and interestingly, and this is one of my favorite topics, everything that has to do with the host and omics, as those that know me better know well, the use of the omic science to study and understand the adverse events or the potential adverse events through the adverse omics. So these and other questions can be answered only if you really are a true expert in the field, and I think with Professor Plotkin, we will be able to cover many of these doubts. But it's not only a question of having a vaccine, to be able to license a vaccine, but to produce them in quantity enough to make them available for all the countries and for all the people that may need it. This is an additional and very important issue to have into account. So this was the introduction I really wanted to do, and now we have the opportunity to discuss about the past lessons and the current lessons from where we can learn in the development of COVID-19 based on the difficult vaccine studies we have been facing in the history of vaccine development. In this picture, you can see, and you can look with envy, a selfie of Professor Plotkin, a selfie of myself with Professor Plotkin. You can understand how proud I can be of having this picture with someone like Professor Plotkin, that has been the inspiration and has been the lead of generations of vaccinologists. He has discovered the rubella vaccines, among other achievements, and he's responsible for saving hundreds of thousands of lives. But he's also the stimulus, and he's the mentor of many, many, many careers in the vaccine field. As said, everybody wants a selfie with Professor Plotkin, and people even annoys us only to be able to appear in the picture. As you can see here, stand some common friends trying to ruin our picture. But for those of you at home that would really like to have a selfie with Stan, you have a much easier opportunity, and you can always get a selfie with Plotkin, because if you are really a person that works in the vaccine field, for sure you will have at your bedside the Plotkin manual, the textbook that anyone that works with vaccines should have near to him as a first aid textbook. So I don't want to steal more time, and now is the time for you, Stan, to talk, and thank you very much again for accepting the invitation. And from here onward, the floor is yours.
So, muchas gracias, Federico. Estoy muy feliz de hablar con todo el mundo aquí y allá. Tenemos muchas vacunas efectivas, pero hay vacunas que carecemos porque el desarrollo es difícil. Hablaré de esas. Es más fácil para mí de hablar en inglés. Dispénsenme. So I'm going to cover a number of different vaccines that we need and that are important. But of course, we live in difficult times because of COVID-19. And so let me begin with that problem. But before going to that, I want to point out something that all of you know very well, and that is that the vaccines that we have, that we use every day, are extremely effective. These are data from the United States, and on the left of the slide, you see the different vaccines. The 20th century morbidity from those diseases before vaccines. And then in the third column, the number of cases in 2016. In 2019, there were even fewer. And lastly, the percentage decrease, showing just how effective in actual use are the vaccines that we have. Next, please. Next slide. Yes. So, how did we develop vaccines in the past? Well, we have, as you know, attenuated vaccines, weakened vaccines. And the first one was Jenner's vaccinia. But then Pasteur attenuated rabies virus and anthrax bacilli. And so that gave us 2 more vaccines. And then with passage of viruses or bacteria in different cell lines, we obtained a number of different vaccines: yellow fever, Bacille Comet-Guerin, And of course, the vaccines we use routinely today, such as measles, varicella, rubella, and other vaccines that are alive, including the rotavirus vaccine that I worked on years ago, and which is protecting infants against infantile diarrhea. The next slide. So, of course, we have also inactivated vaccines. In the late part of the 19th century, different scientists, particularly in England and in Germany, realized that they could kill organisms, but use the killed organisms as vaccines. So, of course, we had killed typhoid, killed a whole cell pertussis vaccine, and going into the 20th century, the inactivated poliovirus vaccine of Salk. But also, we learned how to use toxins, for example, for tetanus vaccine. And we realized in the 20th century that capsular polysaccharides of bacteria could be used as vaccines. And if we conjugated those with proteins, they became even more effective. So that was the past. Next slide, please. Next. Hello? Oops. Next. There we go. So, but the important thing to know is that now we have more strategies for vaccine discovery, as we will discuss in some detail in a moment. But let me point out some of the strategies that we can now use. For example, for attenuated vaccines, we can induce temperature-sensitive mutations, which means changing the temperature at which a virus can grow and attenuate it in that way. Reassortment actually was used for one of the rotavirus vaccines. That is, putting together genetic material from two different sources to obtain a reassortant, which is attenuated. And also viral recombinants and deletion mutants, in other words, removing part of the genetic material to weaken an agent. And then there's something which is quite interesting, and that is called codon deoptimization. By that is meant that you take the genetic code of an agent and you change the code so that the amino acids being coded for are different. And in many cases, that can attenuate an organism. And also people are experimenting with insertion of microRNAs to attenuate and using replication vectors, as I will mention. Next slide. On the inactivated vaccine sides, I will tell you a bit about the use of DNA plasmids as vaccines or messenger RNA. Also, there is something now called reverse vaccinology. And what that is basically is to obtain the genetic code of an organism, and in particular bacteria, and to use bits of the genetic material to immunize mice, for example, and to determine which of the responses that the mice have are protective against the agent that you are trying to develop a vaccine against. And this has been used, for example, to develop one of the current meningococcal B vaccines. Also, we are identifying important antigens of bacteria by transcriptomics, that is, by seeing what is transcribed by the genetic material, and also analyzing the proteins. Structural analysis has been important, as I will show you in one example. And also we'll talk some about adjuvants. Next slide, please. So let me just say a bit more about the approaches using DNA and RNA. What this slide shows is the speed with which one can develop vaccines based on the code of a DNA plasmid. So this is actually relates to the Zika virus, for which we probably do need a vaccine to protect pregnant women. But in any case, the company called Inovio obtained the genetic code for Zika virus on, you can see, November 20th, 2015. And they made a DNA plasmid coding for the virus. Now, the virus is RNA, but they translated the RNA to DNA, and then they used that as a vaccine first in mice, then in monkeys, and their first clinical study was launched in June of the following year. So for vaccine development, that's amazingly fast. And indeed, they did show that their vaccine could protect. So that's what a DNA vaccine could do. Next slide, please. So it was also discovered more recently that you could, in fact, take the genetic code of an agent, and instead of translating it to DNA, you could use the RNA as the vaccine. Now, this is called messenger RNA. There actually is another way of making RNA vaccines, but I won't talk about that. But so this was an effort to develop a Zika RNA vaccine. And although it says DNA in the top, but it was an RNA vaccine. And it did induce antibodies against the virus. So an example of using messenger RNA coding for the Zika virus to induce an immune response. And the responses indeed were excellent in 100% of volunteers. Next, please. Next slide. All right, so now let's talk about COVID-19. Now, one point about nomenclature. I actually prefer to call the virus SARS-2, and the reason is given here. The coronaviruses, or at least the non-respiratory coronaviruses, are parasites of bats, and in particular, it seems, in China. And so from time to time, the viruses from bats can infect mammals. And we don't know exactly how that occurs. It may have occurred in markets where animals were being slaughtered for consumption by humans, but we don't know that for sure. Nevertheless, the first SARS, which was epidemic in 2002, 2003, infected a mammal called civet cats. And as you may recall, that was a highly virulent virus, which fortunately was controlled by isolation. Nevertheless, experimental vaccines were developed against the first SARS virus. And it's important to note that although they were not tested in humans, they did show efficacy in animals. And that is important with regard to the virus that we're coping with now. The second point is that bats have also infected camels. And those who are in contact with camels, such as camel herders, can be infected and are infected, for example, in Saudi Arabia. And they, once infected, those people can transmit to other people. So it's a reasonable question to ask, well, are there vaccines against a Middle Eastern respiratory virus? And the answer is yes. There are a number of vaccines in clinical trials now, and they appear to be effective. So that is good news because it shows that two coronaviruses have been the subject of vaccine development before the current one. So the current one may be coming from a infected pangolin, a small mammal that is eaten by people in China, some people. But in any case, that virus, which I call SARS-2, has, as everybody knows, become epidemic. But I want to stress that the genetic code of SARS-1 and SARS-2 are similar. Not the same, but they're similar, indicating the relationship between the two viruses. Next slide. So let me then talk about what happens and the importance of certain events in coronavirus infection. So this cartoon shows the coronavirus with on the outside these, my picture is apparently obscuring some of the legend, but on the outside of the virus is the spike protein. And the inside, the sort of squiggly line there, is the RNA genome of the virus. And so when someone picks up the virus, let's say, enters the nasopharynx, and it attaches to epithelial cells in the nasopharynx using an angiotensin receptor, ACE2 called, and that enables the virus to enter cells. And of course, the outside protein is broken down, the RNA is released, and the virus is then able to reproduce in the host. So what we know from here then is the importance of the spike protein as the way the virus gets into the cell. Next. So what happens then? Well, the infection is apparently primarily by respiratory droplets, although, of course, contact between people is also highly suspect. But then, as I've said, replication occurs in the upper respiratory tract. And that infection may be asymptomatic, but it's important to note that the way the virus transmits from one person to the other is from the upper respiratory tract. And I stress that because vaccines, as a generalization, do not always prevent infection in the upper airway. They are very good at preventing infections from traveling to organs in the body, such as the lungs. And in this case, that is what we need to block, because in the usual infection, the virus passes from the upper respiratory tract to the lower respiratory tract, and using the same receptor is able to enter the cell. And of course, as you know, cause pneumonia, pulmonary edema, etc. I think it's worth mentioning that the infection also results in activation of T cells. And that activation leads to a cytokine storm and frequently death. And that is important because we know relatively little about the T cell responses to this virus, and whether there are protective T cell responses or whether there are dangerous T cell responses. Next slide. So here is a cartoon of the spike protein. And what I want to emphasize first is on the left part of the structure, where it says RBD. That stands for receptor binding domain. And that, as I will show you in the next slide, is the way the virus actually attaches to the receptor. So that obviously, please go back. And that is in the first part of the spike protein, so-called S1. On the right-hand side, the S2 part is the part that allows the virus to fuse with the membrane of the cell, and therefore to obtain entrance once it's attached to the RBD. So now the next slide shows that the receptor binding domain is at the top of the spike protein, and that is how the virus attaches. And certainly in making a vaccine, we want the vaccine to make antibodies against that domain. Next slide. So what should we choose as the vaccine antigen? Well, one could take the whole virus and simply inactivate it. The concern there is that we might induce responses, particularly to proteins other than the spike protein, which might be not a good idea. It might cause enhancing antibodies that would make it easier for the virus to obtain entrance to the cell. And I've already told you that the spike protein, and particularly the receptor binding domain, are antigens that we definitely want in a vaccine. So the point is, we should use those proteins preferably as vaccines because we don't know exactly what the responses would be to the other parts of the virus. Next slide. So this is, first of all, an overall summary of the vaccines that are being developed. Well, first of all, simply killing the virus and activating it or attempting to attenuate it. There are a few companies doing that. There are many companies, as I'll show you, that are using what we call vectors, and I'll explain that, to make vaccines. And then there are companies using the DNA and RNA approaches that I mentioned to vaccinate, and they are actually in advanced clinical trial at the moment. And then there are other organizations simply using, for example, the spike protein as the vaccine, or making virus-like particles from the spike protein. Next slide. So this gives you more specific information. A DNA vaccine is in clinical trial made by a company actually in Philadelphia called Inovio, and they are in phase 2 now. The difficulty with DNA plasmid vaccines, however, is that one has to use electroporation to get the DNA plasmid through the skin and allow it to attain entry to dendritic cells, etc. But nevertheless, that could be a useful strategy. As far as messenger RNA is concerned, two companies, one in Germany, CureVac, and one in the US, Moderna, have both produced messenger RNA vaccines, and Moderna is also in phase 2 and apparently progressing well. The Imperial College in London is using another approach to RNA vaccine, so-called self-amplifying RNA, but they're not yet in clinical trial. Now, a Chinese company, Sinovac, has actually made an inactivated vaccine, a SARS-2 vaccine, and they are immunizing in China, and it will be very important to see what their results are and whether they obtain good responses that are protective, but also we'll have to be looking for responses that might be enhancing. And then, interestingly, there's a company called Codagenics in the US that is collaborating with the Serum Institute of India. And Codagenics has changed the code of the SARS-2 virus to weaken it, to attenuate it. Now, that has not yet been in humans, but it will be very interesting to see. And then there are non-replicating vectors. Now, let me define what a vector is. A vector is using usually a virus, although it can be a bacteria, and taking out some of the genetic material of that virus, for example, vaccinia virus, and inserting the genetic material for something that you're interested in, as in this case, the spike protein of the coronavirus. And so you have the Janssen company using adenovirus 26 as the vector. You have a Chinese company using adeno 5 as a vector, but adeno 5 infects humans, and that therefore may be a problem in terms of interfering with a response. Oxford University is now far advanced using a chimpanzee adenovirus as vector. Then we have companies using the spike protein, which is inoculated with various adjuvants in various ways. And you can see that GlaxoSmithKline and Sanofi Pasteur are collaborating actually on a subunit vaccine. Novavax is making a virus-like particle from the spike, using the spike protein. And the University of Queensland in Australia has a method whereby the protein is prevented, or how can the synthesis of the protein is done in such a way that the immunogenicity is better than simply producing it without their discovery, which is called a molecular clamp. Then, interestingly, there is a live measles vector vaccine, where the vector is the measles vaccine virus developed at the Institut Pasteur, and they a company called Themis is developing that vaccine. And then lastly, there's vesicular stomatitis virus as a vector, and that goes back to the Ebola vaccine story. So they're using the same vector as was successful for Ebola, but using it with the spike protein. Next. So in summary of the coronavirus situation, we have multiple, over 100 attempts to develop a vaccine. And I am reasonably confident that a vaccine will be developed, but there can be surprises. And one of the major things that we have to look for is the possibility of enhancement as well as protection. So let's talk now about influenza. Next. So, we have both live and inactivated vaccines against influenza. But the inactivated vaccine is not terribly effective in children and in the elderly. And the live vaccine, unfortunately, because the elderly have been infected before, is ineffective. And both types, as you know, are less effective if the virus is changed. And if we're honest, vaccine efficacy is only moderate at best. Although even a 50% effective vaccine has a tremendous epidemiologic effect. So the vaccines, of course, are worth taking, but they need improvement. Next. Next slide. Whoop. Please go back. There was a slide in between there. Well, so the influenza virus has on the outside, like the coronavirus, there it is, what's called the hemagglutinin, as you well know. And the hemagglutinin has two parts to it. The head, shown here in red, and the stalk, the stem, which is shown in blue. Now, it's the head that changes from year to year because of mutations that occur and because of the effect on the head from antibodies that people develop during infection. And it was discovered that although the head is constantly changing, the stalk is not. So obviously, the idea of using the stalk as a vaccine antigen has come to the fore. Next. So the obvious way of trying to get a response to the stalk is shown on the bottom of the slide. That is, you simply take off the head of the hemagglutinin and you use the stalk. However, it was discovered that when that was done, that the conformation of the stalk changed somewhat by taking off the head. And the responses to the stalk were not terribly good. But on the top is another approach which does look promising. And that is using two different influenza viruses. First, you use, let's say, the influenza virus with H5 hemagglutinin, which does not usually circulate in humans. And then you follow with a second injection, let's say using a virus that has hemagglutinin 8 as the head. And what happens is, because you have exposed the stalk in the right conformation after the first injection, and then come again with a second injection, you get good responses to the stalk. Now, this may be very useful in giving a wider immune response to influenza vaccines. Next. Next slide, yes. One other approach is to add influenza virus neuraminidase, which is part of the virus, which allows the virus to leave the cell once it's infected the cell, and has never really been used as a vaccine or part of a vaccine. But I think that should be included in our vaccines. Next. Well, so I've missed a slide here, but let me point out several advanced developments of possibly universal influenza vaccines. One by GlaxoSmithKline, which is using what's called chimeric hemagglutinin. That is to say, taking all of the past hemagglutinins and looking for bits that are important from each hemagglutinin, and putting together those that were at least partially effective in prior years. So that vaccine may be an improvement. Sanofi Pasteur is using something which is computationally optimized antigens. That is, looking at the past influenza viruses and using computer analysis to find those parts which appear to give broad responses. And that is also in trial. And finally, an Israeli company has put together parts of the influenza virus, not only the hemagglutinin, but other parts, and are in phase 2 using that possibly universal influenza vaccine. Next slide. So let me move on to cytomegalovirus. Cytomegalovirus is a ubiquitous virus, practically at least 60%, and in many countries, almost 100% of people are infected with cytomegalovirus, or CMV, as it's called. Next. The interesting thing about CMV, or there are several interesting things, but one that is important, certainly to physicians, is that the virus usually circulates in infants and toddlers in particular, who are, let's say, infected from other infants during the second year of life frequently. And they then can infect their mothers. Now, their mothers may be seronegative to CMV or seropositive. The important thing to realize is that the virus rapidly obtains entry into cells, particularly monocytes, and they circulate, and they can infect the placenta if the woman is pregnant, and the virus therefore can pass to the fetus. Importantly, the same thing can happen in seropositive women, although fortunately at a much lower rate. But it can happen because antibody is not the only protective factor. And therefore, the virus can get into the cells of seropositive women, and therefore, on occasion, infect the fetus. So we have the number one fetal infection that causes damage later. The children infected with CMV are frequently deaf, but they can have much more severe abnormalities, including mental retardation. Next slide. Next. Yeah. So years ago, we developed an attenuated virus, and that is not being developed further at the moment. But the important thing is that we showed that you could protect against CMV in renal transplant patients. What this slide shows is renal transplant patients who were negative for CMV, but who received a kidney from a seropositive donor. And in that situation, as shown in the placebo group in the center, about 35% developed serious CMV disease. But in the vaccinated group, only 5% did so. So it is possible then to protect against CMV, but the question is how to protect pregnant women. Next slide. So this is a cartoon of the virus. And the important thing that I want to show you is on the outside, at the top of the cartoon, is a protein called GB. And next to it, further right, is something called GHGL-UL-128-131, which more simply is called the pentamer because it consists of 5 proteins. And the importance of this is that those 2 proteins or protein constructs are responsible for the antibody responses, particularly neutralizing antibody responses to the virus. Next. Well, I'll pass this slide, which simply, again, shows the protection that you can induce in transplant patients. But a group of scientists at the University of Pavia in Italy have studied pregnant women to determine what are the factors that protect against fetal CMV transmission by following the women during pregnancy. And they found that antibodies to the pentamer are extremely important in preventing transmission to the fetus. Not surprisingly, lower viral load is protective. And they also found that CD4 responses and CD8 responses also correlate with protection against fetal transmission. So here we have a situation where all arms of the immune system are important in protecting against transmission to the fetus. Next slide. So there are now many different candidates and in active clinical trial. There is a replication defective virus made by Merck, and this gives a partial infection. In other words, the virus is able to make a lot of proteins but cannot really reproduce. And this is in phase 2 clinical trial. There are also an attempt to develop virus-like particles, and soluble proteins are being used, for example, by Sanofi, to try to develop a vaccine based on some of the key proteins that I mentioned. And then finally, and again, messenger RNA and DNA plasmids are both being tried as vaccines against fetal infection with CMV. And it will be, in particular, the messenger RNA trials are far advanced. And it will be, of course, extremely interesting to see what the result is. Next slide. So I'm going to skip this. And Federico, ¿cuánto tiempo más? Shall I just...
Please go ahead. All the time you need. I don't know.
Okay. So let me talk about respiratory syncytial virus. You all know that respiratory syncytial virus is the major infection, respiratory infection of children, but also is important in the elderly. And it's a virus for which we need a vaccine, but we don't have one. Next. Well, I've already said this, except for one point, which is that there was a vaccine developed many years ago, which was simply an inactivated, a killed virus. And the problem with that was that it actually, when it was used to immunize children, it actually made respiratory syncytial virus disease worse when they were exposed to RSV naturally. And so that, of course, has made people worry about experimental vaccines for RSV. Next. Next slide. Can we go back to... Um, well, okay, let's move on. Next. Um, so, what are the important proteins of the respiratory syncytial virus? Well, the one that seems to induce the most neutralizing antibody, there are two, but the one that seems to induce most neutralizing antibody is called F for fusion protein. Now, people have tried to use the F protein as a vaccine in the past, but the responses have not been that great and disappointing, and the vaccines did not give much efficacy. But then at National Institutes of Health, it was discovered using structural analysis that the F protein actually changed once it infected cells. That in the center of the slide, you see the sort of round protein prefusion form. That's the way it exists on the virus. But once the virus gets into the cells, shown on the left of the slide, it becomes that sort of Eiffel Tower kind of construct. And the problem was that when people were making the F protein in the laboratory, they were harvesting it from infected cells, and therefore they were getting the post-fusion F on the left. But what they really needed was the prefusion form, because the prefusion form, as shown in the cartoon on the right, has multiple epitopes that induce neutralizing antibody. And so what was needed was to stabilize the F protein. And that was done at NIH. And now there are several trials being conducted of the prefusion form of the virus as a potential vaccine. Next. Next slide. Yeah. Well, I won't go through this slide. Simply shows that in the cotton rat model that antibodies against the prefusion form were protective, which is shown in on the right part in the box, but the post-fusion form was not. Next slide. So now there are multiple candidates for respiratory syncytial virus vaccine, and I won't dwell on each of these except to say that there are attempts to use attenuated RSV. There have been multiple attempts, and those are continuing. There are also vector-based vaccines, but potential vaccines. And it's important to note that a company called Novavax has made a nanoparticle vaccine, a virus-like particle vaccine, that has shown some efficacy in clinical trial. And that vaccine is being developed. It's not yet available, but they hope to develop it into a licensed vaccine. Of course, antibodies against RSV are also being used. Next. So just let me mention, as I will try to close, that in virology now, in vaccinology, we need new adjuvants. Aluminum salts are not sufficient for all needs. Next. And although, can we go back? Um, although, well, all right. The advantage of many adjuvants is that they stimulate what are called pathogen recognition receptors or toll-like receptors. And they come in a number of different receptors, ranging as on this slide. We're including from TLR2 to TLR9. And the point of this slide is that different adjuvants stimulate different pathogen recognition receptors. And some of them are very helpful for protein vaccines. Some of them are helpful for lipopolysaccharide vaccines and so forth. So that vaccinologists now have a whole group of adjuvants that they can use to attempt to increase the immunogenicity of vaccine proteins. And in particular, TLR4 has been used a lot. That is adjuvants that stimulate TLR4. And TLR9 has been used, for example, for a hepatitis B vaccine that is much more immunogenic than the ordinary vaccine. And so this is a very important advance in vaccinology. Next. No, let's pass that one. Keep going. All right, so well, can we go back one? Let me conclude by indicating what I regard as the unsolved problems in vaccinology. General problems. One is that we need to know how best to stimulate immune memory. It's important not only that vaccines protect, but that they protect for long periods of time. For example, the acellular pertussis vaccine is deficient in that respect. It does not produce, they do not produce long-lasting responses. So that's one problem. Another problem is in bacteria in particular, the multiplicity of virulence antigens. And we need better information about those and something called antigenomics, which is the analysis of natural immune responses to a pathogen, to a bacterial pathogen, help us identify which of the antigens are important for in virulence and therefore for vaccines. People have different HLA types, and sometimes to a vaccine there are good responders and bad responders. And so we need vaccines that will stimulate the epitopes that will stimulate responses in a wide variety of people with different HLA types. I mentioned structural biology, identification of conserved epitopes, as in RSV. Systems biology, which allows one to identify which immune responses are important to protection, and therefore to identify the correlate of protection. And for example, it appears that antigens that stimulate interferon may be useful for influenza vaccines. And the next slide, I think the last slide. We don't have yet good ways to stimulate immunity in newborns, and certainly not in the elderly. So how can we do that? So the question is, should we add certain cytokines or neutralize certain cytokines to improve responses in particular groups? That's very much on the table. And then there's the issue of stimulating mucosal responses. We don't have good ways of doing that with vaccines that are not replicating agents. But so-called nanoemulsions are being tried to try to induce responses in the nasopharynx. I've mentioned the issue of adjuvants, and including the importance of adjuvants that generate T-cell immunity without necessarily being replicating vaccines. So, ladies and gentlemen, what I've tried to tell you is that vaccinology is a very active science, that many different ways are being used to apply molecular biology to vaccine development, rather than simply blindly trying to develop vaccines. The world, as we know, is a dangerous place. There will continue to be new agents that will appear. That has happened since human civilization started and will continue to happen. So we need ways of developing vaccines rapidly. And indeed, one of the things that I've proposed and others have proposed is that we start making vaccines against agents that are not necessarily causing disease at the moment, but rather prepare for the possibility that they may become epidemic. But that is something for the future. And so I will stop there, and muchas gracias.
Thank you very much, Stan, for your fascinating talk, and I think we all have enjoyed, and thank you so much for all the discussion you have brought and the many answers you have provided to many of the questions we might have in mind, but also you have elicited also some other doubts. We have several questions from the audience, and I will try to share with you some of them, or all of them if we have time enough, although there are quite many. One of the first is about, in the case we have a COVID-19 vaccine, which is the target population and which is the minimum efficacy we should expect of such a vaccine, and if we should expect to have an annual shot like we do with flu.
Yes, well, very good questions. I'm sorry, the first part of the question was the...
Yes, which is the target population? Whom should receive the vaccine?
Target population. Well, actually, the target population, I would say, is very large. It's essentially everybody. We, of course, if I may express prejudice, we want to protect the elderly who are most likely to die, but we also want to prevent circulation of the virus. And so I think eventually a SARS-2 vaccine, unless the virus disappears, which I doubt, will be used in everybody, including children, because they may very well be vectors for the virus. But obviously, the first target will be healthcare workers. And the second part of the question was...
efficacy we should expect for a vaccine. Yeah.
Minimum
efficacy we should expect.
Yeah. I think if we have a 70 to 80% effective vaccine, the epidemiologic effect will be sufficient to protect the rest of the population. Of course, one would hope for higher efficacy, but we'll have to see. And the point that I mentioned about mucosal replication is, I think, extremely important. I would hope that a vaccine would prevent nasopharyngeal infection, but we'll have to see about that.
And there was
And
another question asking if you think or you foresee that we will need an annual shot like we do with flu.
well, I hope not, but the people who work with respiratory coronaviruses are concerned about that because they have shown that people can be reinfected with the respiratory corona. That being said, when the people were reinfected, they were not symptomatic, so they were protected against disease. And the same thing might be true for another type of coronavirus vaccine. But certainly, we will have to see how long the responses occur before we can make a decision about how often to vaccinate.
Okay, we have also some $1 million questions like, when do you think the first vaccine will be available? And another one is, which will be the winning strategy to develop the SARS-CoV-2 vaccine? So $2 million actually questions.
Well, it's been often said that a vaccine will be available or may be available the end of this year or the beginning of next year. And that's a reasonable generalization, but it's just a guess. We really don't know. And once a vaccine is shown to work, it'll have to be produced in large quantity. And that's something that's never been done before. That is, there are 7 billion people in the world. And so, in principle, we need 7 billion or maybe 2 doses, 14 billion doses. That really is going to be incredibly difficult. I don't have time to talk about this in detail, but I've been one of the people that have been pressing for human challenge experiments to try to speed up the ascertainment of vaccine efficacy. That would be done in young, healthy people and using small amounts of virus. But I don't have time to go into that. If that's not done, then of course the vaccine has to go through phase 1, 2, 3. It's going to depend on a lot of factors, including where the disease is occurring. I don't think you could do such a study in Europe anymore, maybe in Brazil, in some states of the US, maybe, where people are stupidly not isolating themselves. But it's going to be difficult because of the multiplicity of vaccines. We're going to need lots of different places to test vaccines.
Thank you, Stan. Well, we have another question here that is directed to me, and I cannot answer because you didn't answer the previous one. They asked me when the vaccine will be available in Spain and if it will be reimbursed. So I don't know, and I only can hope that we can get at the same time as the rest of the world. That's my best bet and hope if that happens soon.
You know, one point that is worth stressing is that I have never seen an effort to develop a vaccine like this. That is, the multiple companies, the large companies, the small companies, all trying to produce a vaccine. And that's never happened before. In English, one would say it's all hands on deck. Todo el mundo sobre el ponte, right?
Yeah.
And this is terrific. And I hope this is something that will happen in the future. But let me say, I am optimistic about there being a vaccine. But it's just that you have to go through certain steps to be sure of safety and efficacy before you can use it.
Thank you. More questions. I have a difficult question. This comes from a good friend, Javier Diez. He says, Can we ensure before getting the vaccine license that the vaccines won't produce enhanced disease?
Mm-hmm. Yes, that's another very good question, because I think if the phenomenon is something that happens frequently, it will be possible in the usual vaccine development to show that. But if it's something that occurs rarely, that's going to be difficult. So the only way we can get around that is by doing animal studies and in vitro uh, studies. In other words, looking for, as part of the antibody development, not only antibodies that protect, but also antibodies that enhance disease. So, what I would say simply is that our analysis of the immune responses is going to have to be very precise, very detailed, um, because in clinical use, we may not be able to detect that until years have passed, such as in the case of dengue. So it's an important question, but on the positive side, I think the evidence for enhancement with this particular virus does not yet exist.
Thank you. In that same, and I think you already mentioned during your talk of the importance of cell immunity and all the gaps we still have on that regard. There are questions, several questions about the other parts of the immune response and how should we deal with them in the vaccine development, from, of course, the cell response, but also the innate immunity, and what do we know or what can we learn from that in the development of a vaccine against COVID?
Yes, well, so definitely we need to look at a wide range of responses. I did not mention, and this goes back to the question of enhancement, that looking at different cellular responses was useful in identifying enhancement in other coronaviruses. As a generalization, one can say that CD4 responses, we really need to look both at CD4 and CD8 responses. CD4 cells, as you know, do multiple things. And one of the things that they do is orient towards Th1, which is what we want, rather than Th2. And importantly, Th17. And this goes back to the point that I raised about nasopharyngeal carriage, because we know that Th17 responses are important in preventing nasopharyngeal colonization. And at this point, we do not really know enough about the vaccines that are being tested to know whether they will or will not induce that type of response. With regard to CD8, all I can say is that I know of no evidence, no concrete evidence that they are important in preventing disease. Obviously, they may be important in controlling infection once it occurs, but in terms of a vaccine, I'm not sure that we will need that. So looking at cellular responses will be important, without doubt, but it will require a fine analysis, not simply ELISpot counts, but functional activity.
Yeah, I can't agree more with that. There are several questions about safety and the compatibility of the speed race we are facing in the development of vaccines against COVID and to the guarantee of safety. We have mentioned already the enhanced, but there are several questions of how reliable this will be, if we can trust the regulatory authorities that will guarantee that, despite the rush, no vaccine won't fulfill the necessary requirements in terms of safety. What's your view in this regard?
Well, so it's not an easy question to answer because, of course, we face a situation where the economies and the civilization of the world are being destroyed. Um, uh, I can no longer go to the Metropolitan Opera in New York because they are not producing anything. But without making a joke of it, the fact that people have lost their jobs is very important. So we need to move as rapidly as possible. But of course, at all stages, we have to be concerned with safety. I have, in my old age, started to do some work on safety. And for example, in the US, analyzing reactions with respect to numbers of doses shows that real reactions to vaccines occur at a rate of about 1 to 2 per million doses. My point is that even the vaccines we have do cause safety problems in some people, but rarely. And I think the same thing is likely to be true with a coronavirus vaccine, that there will be reactions that we will have to look for. But I do think that we can guarantee overall safety. That is, that that in a large number of people, perhaps in the range of 5 to 10,000, that we don't see safety problems. Later on, of course, we'll have to have observations of safety to continue. But what I'm saying is that we cannot allow safety issues to slow vaccine development. Rather, we need to include as much safety observations as possible during vaccine development and test in phase 3 as 5 to 10,000 people, and then continue observations. But if we have something that's safe in 5 or 10,000 people, it is unlikely that it's going to be dangerous for the vast majority of people who need the vaccine.
Thank you. I have some other questions that are related to the other vaccines, those vaccines that are already available. I also mentioned during my talk the role or the potential role of co-infection, and I mentioned also the role that routine vaccination and the maintenance of routine vaccination might have. But there are questions specifically asking for the potential cross-protection of flu vaccine or pneumococcal conjugate vaccine. What's your view on that?
Well, you mean the non-specific effects of vaccines?
No, well, the questions are several. I mean, there might be the non-specific ones, but they are also talking about other routine vaccines like flu or pneumococcal conjugate vaccine, if they might be protective in COVID patients.
Oh, I see. That's also, yes, I should have understood the question. I have been talking to people and actually involved with a project just on that subject because, I mean, non-specific effects is not probably a good term, but we know that measles vaccine, for example, can influence other types of disease and be protective. And it was discovered, for example, that there is homology between a so-called P150 protein of rubella virus and one of the non-structural proteins of SARS-2. And so people are asking, well, should MMR be used to give some protection against SARS-2? Well, of course, the answer is we don't know whether that would be true or not. But I think it is beyond doubt that vaccinations not only have an effect against the target disease, but it changes the immune system. And fortunately, as far as I can tell, mostly for the better. And therefore, it is possible that other types of stimulus might help protect. One thing I think is related to the point that we don't know why some people in the right age group die and some people in that same age group aren't sick at all. Why is that? We simply don't know. And it may be because they have had some other infections which are giving them protection. One of my neighbors, his family was thoroughly infected with SARS-2, his wife, his children, and he was infected but totally asymptomatic. Why? Nobody knows.
Yeah, absolutely. That's where the host also has something to say, and we have to look also at the host perspective. That's precisely
Yes.
what the research approach I like more. I think that, I mean, we are out of time and we have a few more questions, but before we finish, I like to make, well, at least one of my own questions. And this is, what do you think about that among all the candidates that are being pursued and all the technologies that are being applied in the search of a COVID-19 vaccine, the more classical, let's say, approach, the inactivated or attenuated ones, are not being currently at all considered in the US or in Europe. No, I mean, all the candidates are in China or the one you mentioned from India. Do you think we are omitting the classical way and we can be getting an unnecessary risk, or what's your view on that?
Uh, it's a difficult question, Federico, because if we knew which vaccines were effective, of course, I could answer the question. For example, if the whole inactivated virus is effective, that's a very easy way to make vaccines, and we could make millions and billions of doses. Uh, I really don't know the answer to that, but what I do like is the fact that every different strategy is being used experimentally to determine which is the best approach. And so we will learn a lot about vaccines from this experience. I am optimistic that we will have effective vaccines and maybe multiple vaccines. But until we have the data, we're simply guessing. Uh, Personally, I would like to receive the messenger RNA vaccine coding for the spike protein. But I will be happy to receive any one of the experimental vaccines.
Okay, thank you so much, Stan, for not only for your magnificent talk, but your I mean inspiring answers to all these questions. It has been a great pleasure to have you with us tonight or today, depending on where from where people is connected. And I really want to thank you once more on behalf of my institution, on behalf of all the entities that have endorsed this activity, my hospital, my service, my research institution, but mainly from myself. It has been really, I'm really grateful for having accepted this challenge and having made possible us to enjoy as much learning as always. We have the chance to listen to you more than we could even expect. Thank you very much, Stan. I also want to thank all the audience for attending the webinar. I hope they have enjoyed it. Also the IT team behind, because so far, or at least up to what I could see, there were no any issue, which is what we can expect from the IT teams. I also want to thank Pfizer for their support to be able to develop this webinar, and well, also to wish the best for all our audience, to remain safe, and while we don't have the COVID vaccines, to get sure that we receive all the other vaccines we know that work, not only for our children, but also for ourselves. As adults, we can receive pneumococcal conjugate vaccination, flu vaccination. There are other reasons to avoid going to the hospital, to avoid getting ill, and these are good reasons to remain safe. So I hope we have fulfilled everybody's expectations with the webinar, and after this, it will be made available online for those of you that want to go through some of the questions or some of the details. Again, Stan, thank you so much. It has been great to have you, even in the despite being in the distance. But I will bring you to Spain as soon as we can do it, as you know.
Con mucho placer.
Thank you very much.
Hasta la vista.
Bye. And thank you very much. Muchas gracias a todos.